The future of medicine, synthetic blood. Imagine a world where blood shortages are a thing of the past, where emergency rooms never have to turn away patients due to lack of compatible blood, where surgeries are safer and lives are saved more efficiently. Sounds like science fiction, right? According to Andrew Zaleski, this future might be closer than you think. From milk transfusions to ErythroMer. Join us on a captivating voyage through history, from the peculiar milk transfusions of the 1800s to the revolutionary advancements in synthetic blood substitutes like ErythroMer. Buckle up, because this is going to be an eye-opening ride. Welcome to a new world that uses synthetic blood. Hey there! If you're new, we're here to help you discover cool new tech gadgets and break down tricky science concepts into easy-to-understand explanations. Today, we're diving into something that's not just techie but also incredibly life-changing: synthetic blood. So, what exactly is synthetic blood? In simple terms, it's an artificial substance designed to mimic and perform some or all functions of natural human blood. Unlike traditional blood transfusions that require matching blood types, synthetic blood aims to be universally compatible and readily available. Now you might be wondering why this topic is relevant for tech enthusiasts like us. Well, synthetic blood isn't just about medical science. It involves cutting-edge technology in biotechnology and nanotechnology. It's a perfect example of how interdisciplinary innovation can solve real-world problems. Before delving into the intricate details of synthetic blood and its future prospects, let's briefly examine its history. Trust me, it's as fascinating as it gets. The science behind synthetic blood. Alright, let's get technical for a moment. Human blood primarily transports oxygen from the lungs to different tissues in the body and carries carbon dioxide back to the lungs to be exhaled. This crucial task is performed by red blood cells containing hemoglobin. Synthetic blood aims to replicate this function using different materials and technologies. There are various approaches that utilize hemoglobin-based oxygen carriers, commonly referred to as box, which are derived from human or animal hemoglobin but have been modified to ensure safety and effectiveness. Others use perfluorocarbon emulsions, which are synthetic compounds capable of dissolving large amounts of gases like oxygen. So how does it work? ErythroMer is a highly promising synthetic blood substitute currently undergoing extensive research. It is designed as a freeze-dried powder that can be mixed with water when needed, making it incredibly convenient for emergency situations where traditional blood storage and transportation are challenging. The main concept behind ErythroMer revolves around encasing hemoglobin within minuscule nanoparticles that can effectively travel through the bloodstream without triggering any negative responses in the patient's body. ErythroMer is a potential game-changer. ErythroMer has the potential to revolutionize the approach to managing medical emergencies characterized by severe bleeding or trauma. Imagine soldiers on the battlefield having instant access to life-saving transfusions, or remote clinics in developing countries being able to treat patients without the need for refrigeration or worrying about finding matching blood donors. Although still in its experimental stages, ErythroMer has demonstrated promising results in preclinical trials involving animals. Its potential is truly exciting. Researchers are optimistic about moving towards human trials soon. Historical attempts, from milk to saline solutions. Back in the 1800s, doctors were desperate for alternatives due to frequent fatalities from excessive bleeding during surgeries or childbirths. Theodore Gallard Thomas's bold experiment with milk transfusions. In 1875, a time long before we had a comprehensive understanding of blood types, a renowned New York gynecologist named Theodore Gallard Thomas tried something radical: injecting cow's milk directly into patients' veins. Yes, you heard that right. Thomas believed milk could serve as an effective replacement due to its nutrient-rich composition, resembling plasma components found naturally within our body's circulatory systems, or so he thought. He administered 175 milliliters, approximately 6 ounces, of cow's milk intravenously. The procedure was performed to treat severe uterine bleeding in a woman who had recently undergone surgery to remove cancerous ovaries. This incredible utilization of cow's milk, equivalent to a small juice box or half a can of soda, took place during the Victorian era when medical techniques were considerably more rudimentary compared to the present day. Despite the difficulties and lack of resources at that time, this innovative approach shows the determination and creativity of those involved in finding healthcare solutions. The patient initially complained of feeling like her head would explode. She also had fever and a rapid heartbeat. However, miraculously, She recovered a week later. Thomas conducted 7 more procedures similar to the ones that were successful. He published the results in medical journals, predicting a promising future for the practice. Despite skepticism from his peers, Thomas remained optimistic. Although saline solutions were introduced as safer alternatives for emergency bleeding and are still widely used today. However, the search for more effective substitutes, like synthetic blood, continues in order to further enhance medical treatment outcomes. ABO blood group system. Although these early attempts might seem rudimentary by modern standards, they laid the groundwork for understanding the importance of compatibility and safety in transfusion practices. This foundation ultimately resulted in the discovery of ABO-RHD grouping systems, which have since saved innumerable lives. The discovery of the ABO blood group system was made by the Austrian scientist Karl Landsteiner in 1900. In 1940, Landsteiner made a groundbreaking discovery in collaboration with Alexander S. Wiener. They identified the Rh blood group system, uncovering the positive or negative blood factor known as the Rh factor. These incredible discoveries have revolutionized blood transfusion practices and saved countless lives since then. The ABO-RHD grouping system is a classification method used to determine the compatibility of human blood types by identifying specific antigens on the surface of red blood cells. The ABO blood system identifies blood types as A, B, AB, or O based on the A and or B antigens found on the cell surface. The term RHD refers to the rhesus D antigen. If this antigen is present, the blood type is considered positive. If it's absent, the blood type is negative. Consequently, we get 8 main blood types: A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. The grouping system plays a crucial role in blood transfusion and organ transplant procedures, as incompatible donations have the potential to provoke detrimental immune reactions. Challenges: ethical considerations. Despite advancements in developing viable substitutes, several challenges remain to be addressed before widespread adoption is possible, including ensuring long-term safety efficacy, preventing immune reactions and clotting issues, scaling production to meet global demands cost-effectively, addressing ethical concerns, sourcing materials, especially animal-derived products, navigating regulatory approvals and ensuring compliance with rigorous testing standards are essential factors in successfully meeting industry requirements. Researchers are hopeful that overcoming these obstacles will lead to a better and healthier future for everyone. Future prospects, innovations, substitutes. Envisioning a promising future. With groundbreaking advancements, venturing into uncharted territories that transcend conventional technological enhancements. Stem cell-derived red cells grown in a lab potentially offering limitless supply tailored to patient-specific needs, reducing risks of incompatibility rejection. Gene editing techniques enhance the properties of natural cells, boosting their performance and durability under extreme conditions. Advanced biomaterials mimicking complex structures and native tissues, enabling seamless integration of artificial components into biological systems. The possibilities are truly endless, only limited by our imagination and the ingenuity of scientists and engineers who work tirelessly to bring these visions to reality, benefiting humanity as a whole. Wow, we've covered a lot of ground today. From bizarre experiments involving cow's milk pioneering efforts, modern breakthroughs like ErythroMer, a potential game-changer in the field of medicine. Let's quickly recap key points discussed. Synthetic aims to replicate natural functions using innovative approaches, materials, and technologies, also known as biotechnology. This field specializes in utilizing living systems and organisms to develop and create a wide range of products. Historical attempts emphasized the significance of compatibility and safety as the foundation for current practices. Despite the challenges and ethical considerations, researchers are optimistic about overcoming them and paving the way for a brighter and healthier future. Despite the difficulties and moral dilemmas surrounding the development of synthetic blood, scientists remain hopeful. They believe that they can surmount these challenges and significantly contribute to improvements in healthcare and the well-being of all stakeholders in this field, including patients, doctors, family members, and biotechnological companies, all of whom have a vested interest in the success of synthetic blood. The future is indeed bright, filled with endless possibilities of innovations transforming landscape healthcare forever. Stay tuned for more exciting updates and breakthroughs coming your way soon. And don't forget, If enjoyed learning about this fascinating topic, make sure subscribe channel, hit like button, and share this video with friends and family that are interested in cutting-edge advancements shaping tomorrow today. Also check out the next video exploring other incredible medical breakthroughs pushing boundaries of what is possible and improving quality life across the globe.